Welcome! This series of tutorials aims to inform diagnostic laboratories about the new regulation on in vitro diagnostic medical devices, the IVDR. Please note that the implementation of the IVDR is an ongoing process and many aspects remain to be clarified. This means that new information will become available continuously. Please visit the ESLO website for more information. 5. Preparatory Action 2. Assay inventory. The second action to prepare for the IVDR is to make an inventory, with the goal to obtain an overview of the tests that you will be running after the date of application, so after May 2022. This will help you with planning the coming year and with defining the device-specific actions that will need to be taken. The best way to do this is by reviewing your current assay portfolio. Start by listing all the assays that are used in your lab and specify which ones are CE-IVDs and which ones are in-house devices. For CE-IVDs, the main question is whether they will be CE marked under the IVDR, as this will be essential for continued use of the test. This is not a straightforward question, and to explain this, we will first look into a few requirements for IVD manufacturers and their CE-IVDs. Most of these requirements also apply to in-house devices, so knowledge of these requirements will also be relevant for those devices. So, let's have a look at some of the most important requirements for manufacturers. The first one is the risk-based classification system. The system that is used under the IVDR is shown in the table. There will be 4 classes, Class A to Class D. Class A is the lowest risk class, low risk for individual patients, and Class D is the highest risk class, high public health risk. For hemato-oncology and immunology laboratories, a lot of assays fall into Class C, high risk for individual patients or a moderate public health risk. These are mainly genetic tests, flow cytometric tests, and other cancer tests. This risk-based classification system applies to manufacturers and laboratories. For each device, they will have to determine in which class it falls. The higher the risk, the stricter the requirements and the need to show that your devices perform as intended. The second one concerns providing clinical evidence. This is well defined in the IVDR. Clinical evidence consists of 3 different parts. Scientific validity, or the association of an analyte with a clinical condition or a physiological state. The proof is often based on literature. Analytical performance, or the ability to correctly detect an analyte. This is usually shown by performing analytical performance studies. And clinical performance, or the ability to yield results relevant for a clinical condition or physiological state. Clinical performance is a new requirement. It can be based on published literature. So, in first instance, a manufacturer will review the literature to see which studies have been performed with their devices. In case the available data is not sufficient to support the clinical performance requirement, they will need to set up new clinical performance studies to collect the required data. This can be a major effort for manufacturers. So, how does a manufacturer or a laboratory know which clinical evidence needs to be presented exactly? This all depends on the intended purpose that they defined for the device. The intended purpose is a central term in the IVDR. It describes the analyte that is measured, the clinical conditions for which the IVD can be used, the age groups, the type of specimen, etc. The clinical evidence should support the full intended purpose. If this is not the case, the intended purpose should be adjusted to fit with the available evidence. With other words, the IVD can only be used for applications that are supported by evidence. Let's have a quick look at this figure, which depicts well how these types of evidence relate to each other. Scientific validity concerns the relation between clinical condition and the analyte. Analytical performance connects the device with the analyte, and clinical performance connects the device and the clinical condition. After a device is marketed, manufacturers will have to continuously update this information as part of a process called post-market surveillance. For Class C and D devices, this needs to be done at least every year. In conclusion, the clinical evidence and post-market surveillance requirements can be very demanding for manufacturers. And the third requirement, the third-party assessment by notified bodies. This one does not apply to in-house devices, as laboratories are only checked by their competent authority and not by a notified body. Notified bodies assess the files of IVD manufacturers. As stated before, under the IVDD, only a number of high-risk devices need to be certified by a notified body. All other devices are self-certified, meaning that when a manufacturer determines that all requirements have been fulfilled and all documentation has been generated, they can say, okay, I am compliant, I CE mark my device and put it on the market. Under the IVDR, almost all devices will have to be certified by a notified body, and only the non-sterile Class A devices can be self-certified. This means that the percentage of IVDs that need to be assessed by a notified body will increase from 20 or even less under the IVDD to 80 or even more under the IVDR. What does this mean for manufacturers? There are stricter requirements that will really take some effort to meet. In addition, there's a much higher involvement of notified bodies, and with a limited number of notified bodies available at this moment, this is really a bottleneck. So, manufacturers will often have to prioritize the devices that they CE mark. This means that some CE IVDs might disappear from the market because they are not profitable enough, or they are relatively low priority. Other CE IVDs might temporarily disappear from the market when a manufacturer is not able to CE mark it on time under the IVDR. Back to the assay inventory. Does a CE IVD stay on the market? This is an important question to ask. It is a good idea to contact your distributors or manufacturers and ask them what their plan is. Do they know already if their devices will be available after May 2022? Based on such inquiries, it turns out that some CE IVDs are discontinued. However, for most CE IVDs, manufacturers say that it is their intention to keep them on the market, but frequently they have not engaged successfully with a notified body yet, and/or do not have a clear timeline. So it is good to keep in touch with distributors and manufacturers on this topic. Of course, if the manufacturer will CE mark the device, you can continue to use it. If it will be discontinued, there are a few alternatives. You can switch to an in-house device, so implement an in-house test to be able to still run your analysis and get the results that you need. This would be an effort, of course. Or you can send your samples to another lab that can do the test for you. Or you can just stop the test altogether. Then let's have a look at the other side, the in-house devices. There are two questions that are important here. Is an equivalent CE IVD available on the market? And if so, does it or do these meet the target patient group's specific needs at the appropriate level of performance? These questions come from Article 5.5d. Note that these terms, equivalent and target patient group's specific needs or appropriate level of performance, are terms that remain to be defined. So at this moment, we don't know exactly how to interpret them. Anyway, if there are no equivalent devices available on the market, you can continue to use your in-house device. If there are equivalent devices available, you will need to assess whether they have the appropriate level of performance. It is often said that a CE IVD should be at least as good as the in-house device. In this case, you will have to use the CE IVD, and you are not allowed to use the in-house device. However, if your in-house device performs better, you can use the in-house device for the application. A few more words on in-house devices and the interpretation of Article 5.5. As mentioned before, Article 5.5 is a brief description of conditions for in-house devices, and various terms in this article are expected to be clarified in the upcoming MDCG guidance document. For example, equivalent and industrial scale. But it is important to realize that this guidance document will only explain these terms up to a certain level of detail. Labs will still need to make decisions by themselves, preferably in a collective way, because every diagnostic specialty has their own assays and their own questions. This means that specialization-specific guidelines will be needed on top of this guidance, preferably developed in alignment with the national competent authorities, as they will be performing the inspections. Then concerning the need to look for equivalent CE IVDs and to compare them with your in-house devices, based on Article 5.5d. Since manufacturers will need to submit information on their medical devices to EUDAMED, the European Databank on Medical Devices, this database will be the designated place to perform such CE IVD monitoring. However, the number of CE IVDs that have been certified under the IVDR is very limited so far, and also EUDAMED is not functional yet. That means that at present, it is not straightforward to search for available alternatives for your in-house devices. Other sources can of course be used, such as literature, company searches, or your expert knowledge. Labs might prefer to still rely on their in-house devices if it is not yet clear that an appropriate alternative is available and will stay available. Finally, modified and off-label CE IVDs and research use only kits, RUO kits, will most likely be considered to be in-house devices, so the same Article 5.5 and Annex 1 requirements would apply to those tests.